So welcome guys. In today's session, we'll be looking at prokaryotic gene transcription, and by the end of this session, you should know uh, RNA polymerase, that is prokaryotes, and also know the steps involved in RNA synthesis, or the steps involved in the prokaryotic gene transcription. So welcome all. My name is Horis Munyiri, and I'm the Dr. Now let's start with this overview. Now, bacteria or prokaryotes, one species of RNA polymerase synthesizes all the RNA. Okay, so only one species of RNA polymerase will be needed to synthesize, be it messenger RNA, transfer RNA, or ribosomal RNA. But we have one RNA which is not synthesized by this RNA polymerase. That is the short RNA primers, which are usually required in DNA replication. Remember, they were being synthesized by the primase. Remember, the primase was DNA-dependent RNA polymerase. Okay, now, and it was a monomeric enzyme. Okay, now RNA polymerase is a multi-subunit enzyme. That is, it has multiple subunits with a molecular weight of about 480 kilodaltons. Okay, and it usually recognizes the promoter region at the beginning of the end of the DNA that is to be transcribed. So you see there, it has a subunit which will be responsible for the recognition of the promoter region. Now, then the RNA polymerase will make a complementary RNA copy of the DNA template strand, and then it will recognize the termination region, and where it will now terminate, it will terminate the synthesis. Okay. Now, RNA is usually synthesized in the 5' 3' direction, and it is anti-parallel to the DNA template strand. Okay. Now, the template is copied as it is in DNA synthesis, in that guanine on DNA will specify a cytosine on the RNA, and a cytosine on the DNA will specify a guanine, and a thymine will specify an adenine, but an adenine will not specify a thymine, rather it will specify a uracil. Okay. Then the RNA is complementary. It is complementary to the DNA template, which is also known as the antisense or the minus strand, but it is identical to the coding, the sense or the plus strand, with only uracil replacing thymine. Okay. Then within a DNA molecule, remember it is it's a duplex. So the regions of both strands can serve as templates for transcription. Okay. So for a given gene, okay, however, only one of the two DNA strands can be used as a template. Now, which strand is used as the template will be determined by the location of the promoter for that gene. Okay. Now, transcription by the RNA polymerase involves a core enzyme, okay, and several auxiliary proteins. Let's look at the structure of the RNA polymerase. Here you can see a core enzyme and a hollow enzyme. Now, the hollow enzyme has 6 subunits. Sigma subunit here, 2 alpha subunits, a beta subunit, a beta prime subunit, and an omega subunit. Remember, this is the uppercase of omega. We have the lowercase of omega, which looks like a small case of the letter W. Okay, so that is the hollow enzyme. Now, the core enzyme refers to the hollow enzyme without the sigma factor. So once the sigma factor recognizes the promoter region, it is usually dropped off. So it usually fall off, and the remaining 5 subunits constitute the core enzyme. So let's look at the functions of this subunit. Now, what is the function of the alpha subunit? It usually determines the DNA region which is going to be transcribed. The beta subunit, this one, usually it has the 5' 3' polymerase activity, and it will catalyze the polymerization. The beta prime here usually bind and open the DNA template, and it has a helicase activity. Okay, then the sigma subunit found in the hollow enzyme, it is the one which will recognize the promoter region for initiation. Okay, for the process of initiation to occur. Now, function of the omega subunit is still not is unknown. Now, The process of transcription of a typical gene in E. coli can be divided into three phases. Have three phases: initiation, elongation, and termination, just like in DNA replication. Now, a transcription unit extends from the promoter to the termination regions, and the initial product of transcription by the RNA polymerase is termed as the primary transcript. Okay. Now, initiation. Transcription begins with the binding of the RNA polymerase holoenzyme to a region of the DNA known as the promoter region, but it is not transcribed. The promoter region is not transcribed. Okay. The RNA polymerase we just bind there. Now, the prokaryotic promoter region contains a characteristic consensus sequence. Okay. These consensus sequences are idealized sequences in which the base shown at each position is the base most frequently, but not always encountered at that position. Okay, so it is the base most frequently observed at that position, but not always. Okay, now those that are recognized by the prokaryotic RNA polymerase sigma factor, the subunit, include the following: the negative 35 sequence, okay, which is a consensus sequence of TT-GACA, TTGACA, which is centered about 35 bases to the left of the transcription start site. So it is located 35 bases upstream of the transcription start site, okay? And it is the initial point of contact for the holoenzyme, and a closed complex is formed. So what do you mean by closed complex? Is that the holoenzyme will just bind there, but it will not unwind the DNA duplex, okay? Then after binding there to the negative 35 sequence, it will slide, it will move downstream and cover the second consensus sequence, the Tata art, or also known as the Pribno box, which is centered about 10 bases upstream of the transcription start site. And once it reaches there, the beta prime subunit will catalyze the unwinding of a short stretch of DNA consisting of about 14 base pairs. Okay, this initial melting will convert now the closed initiation complex to an open complex. Okay, so you can see when it was at the negative 35 sequence, there was a closed complex, but once it reaches the Pribno box, open complex is formed, and that open complex is known as now the transcription bubble. This is what I mean. You can see here, this is a transcription start site, skip one. Now, all bases which are located to the left of the transcription start site or upstream, they are denoted as negative, with the negatives. So at a region found about 35 bases to the left of the transcription start site is known as the negative 35 sequence, the TT-GACA, and this is where the holoenzyme will first bind and form a closed complex. Then it will slide downstream until the Pribno box, where now the beta prime subunit will catalyze the unwinding of the DNA duplex to form an open complex, the transcription bubble. Now, elongation, how does this occur? Once the promoter has been recognized and bound by the holoenzyme, local unwinding of the DNA helix continues, and this is mediated by the RNA polymerase. Okay. Now, unwinding will generate supercoils in the DNA that are usually relieved by the DNA topoisomerases. Remember the two types: DNA topoisomerase 1, DNA topoisomerase 2. Remember, especially we had a type of DNA topoisomerase 2 found in bacteria, the DNA gyrase. And I want you to remember one class of drugs which was inhibiting the DNA gyrase known as the fluoroquinolones, an example being ciprofloxacin. Okay. Now, RNA polymerase begins to synthesize a transcript of the DNA sequence, and several short pieces of RNA are made and discarded. Okay. Now, the elongation phase will begin when the transcript, which usually start with a purine, especially GTP. Okay. Remember, purine is 1,9 adenine, so it will mostly start with a G. Okay. Now, the elongation phase now will begin when the transcript exceeds about 10 nucleotides in length, and the sigma subunit is then released from the holoenzyme to form now the coenzyme, which is now able to clear the promoter region. This is known as promoter clearance, and move along the template strand in a processive manner, serving as its own sliding clamp. Okay, so the RNA polymerase serves as its own sliding clamp. Remember, in DNA replication, like in DNA polymerase in prokaryotes, the beta subunits were serving as the sliding clamps, and in eukaryotic DNA polymerase, the proliferating cell nuclear antigen is the one which was serving as the sliding clamp for the enzyme. But here, the RNA polymerase is serving as its own sliding clamp. Now, during transcription, a short DNA-RNA hybrid helix is usually formed. Now, what are some similarities between DNA polymerase and RNA polymerase? Like in DNA polymerase, it will also utilize nucleoside triphosphate as the substrate and release a pyrophosphate each time a nucleoside monophosphate is added to the gluing chain at the 3' end. Now, as with also DNA replication, transcription occurs in the 5' to 3' direction. But in contrast to DNA polymerase, RNA polymerase does not require a primer. So there is no function, there is no need for a primase, okay? And it does not have the 3', 5' exonuclease activity for proofreading. However, that does not mean that it does not proofread. When we have a misincorporation of a ribonucleotide, the RNA polymerase usually pause, okay, backtrack, okay, cleave the transcript it had created, and then restart. Nonetheless, even if it does that, transcription has a higher error rate than replication does. So this is what I'm saying here. You can see the RNA polymerase. You can see here the RNA being made here, and you can see here the RNA DNA hybrid helix. And remember, this will proceed, elongation will proceed in the 5', 3' direction, but the RNA polymerase will move towards the 3' end of the non-template strand, but towards the 5' end of the template strand. Now, termination. Once you have elongated, you need to terminate. Now, the elongation of the single-stranded RNA chain continues until a termination signal is reached. Now, termination can occur in 2 ways. It can be intrinsic, also known as raw independent, that it occurs without additional proteins, or dependent upon a participation of a protein known as the raw factor. So one is raw independent or the intrinsic termination, and the other one is raw dependent termination. How does the raw independent termination occur? It is seen in most prokaryotic genes, and this will require that a sequence in the DNA template will generate a sequence in the nascent RNA that is self-complementary. Now, this self-complementary sequence will allow the RNA to fold back on itself, forming a GC-rich stem. Remember, the bonds between G and C are 3 hydrogen bonds, so that GC-rich stem will be stabilized by those hydrogen bonds plus a loop. This will form a structure known as a hairpin. Okay. Additionally, just beyond the hairpin, the RNA transcript will contain a string of uracids at the 3' end. And remember the bonding between these uracids and the complementary adenines of the DNA template is weak because you have 2 hydrogen bonds. And due to that weak bond, those weak bonds, that will facilitate the separation of the newly synthesized RNA from the DNA template as the double helix crosses behind the RNA polymerase, or the zips up behind the RNA polymerase. This is what I'm saying. Here you can see in the DNA template strand, this region here and this region here are self-complementary, such that the nascent RNA formed can fold back on itself. Let me take a pen and show you that, what I'm saying. You can see this C here can bind with this G here, and this C here can also bind with this G here, as shown here, and they will form this hairpin. They will form this hairpin here structure. Then, just beyond the hairpin, we have this a string of U's, and this will form weak. They have weak bond binding with the complementary adenine. So 2 hydrogen bonds, and like here you have 3. So this weak binding will allow the release of the nascent RNA. Then how does the, now let's look at the other one. How does the other one, the rho dependent termination occur? It will require the participation of an additional protein known as the rho factor, which is a hexameric ATPase with helicase activity. Now, the rho factor usually binds to a C-rich rho utilization site near the 5' end of the nascent RNA, and then using its ATPase activity, will move along the RNA until it reaches the RNA polymerase, which is posed at the termination site. Then, using the ATP-dependent helicase activity, the rho factor will separate the RNA-DNA hybrid helix, causing the release of the RNA. Okay, then let's look at some clinical application of this prokaryotic RNA, prokaryotic gene transcription. We have some antibiotics which prevent bacterial cell growth by inhibiting RNA synthesis. One good example is rifampicin, which inhibits transcription initiation by binding to the beta subunit of the prokaryotic RNA polymerase. And by doing so, it will prevent chain growth. It will prevent chain growth beyond the 3 nucleotides. Okay, rifampicin is important in the treatment of tuberculosis. So it will bind to the RNA polymerase of it will bind to the beta subunit of the RNA polymerase of Mycobacterium tuberculosis, and by doing so, it will prevent bacterial cell growth and can be used in the treatment of TB. Then we have another drug which is known as DACTINOMYCIN or actinomycin D, which has slightly a different mechanism of action in that it was the first antibiotic to find therapeutic application in tumor chemotherapy. How does it work? It usually intercalates or insert between the DNA bases, and by doing so, it will inhibit transcription initiation and elongation. Now, here you can see this is the RNA polymerase. Okay, rifampicin will bind to the beta subunit here, and by doing so, it will prevent chain growth beyond the 3 nucleotides. You can just a very short RNA synthesis. Cannot grow beyond the 3 nucleotides. Okay, and does rifampicin have an effect on the mammalian or the eukaryotic RNA polymerase? Yes, it can bind to the mammalian RNA polymerase, but if the drug is maintained within the therapeutic index, it has no toxicity on the mammalian RNA polymerase. So the RNA polymerase of the host will not be affected, but that of the prokaryote, let's say the Mycobacterium tuberculosis within the host, it will be severely affected. Okay, so I think we have come to the end, and I want to thank you guys for your attention, and subscribe for more anatomy and biochemistry content. Thank you.